At the end of meiosis 1, we have two haploid daughter cells. When meiosis 2 begins, the chromosomes still consist of duplicate chromatids. By the end of this presentation, you will be able to identify how many sets of genes are found in most adult organisms, describe the events that occur during each phase of meiosis, identify the differences between meiosis and mitosis, and explain how two alleles from different genes can be inherited together. Look at the two cells at the top of the figure. The cells have two one-end chromosomes, but four chromatids. Unlike the first division, neither cell goes through a round of chromosome replication before entering meiosis 2. During metaphase of meiosis 2, chromosomes line up in the center of each cell. As the cell enters anaphase, the paired chromatids separate. The final four phases of meiosis 2 are similar to those in meiosis 1. However, the result is four haploid daughter cells. In anaphase of mitosis and in anaphase 2 of meiosis, sister chromatids separate. But in anaphase 2, there are no homologous chromosomes. The cell is haploid, not diploid. Therefore, anaphase 2 of meiosis is somewhat different from anaphase of mitosis. The chromosome number of each of these cells is 2. Remember that the chromosome number of the original parent cell was 4. We get 4 haploid daughter cells at the end of meiosis 2. Look at the slide and predict the terms that best complete the sentence. The end product of meiosis is 4 daughter cells that have half the normal chromosome number. They are haploid, or one-end cells, instead of diploid, or two-end cells. You can see that meiosis resulted in cells with half the normal chromosome number. This is important for gametes because then when gametes fuse, when one gamete fertilizes another, then they will return to having the normal number of chromosomes. You will get from 2 haploid cells, you will get 1 diploid cell. Some differences between meiosis 1 and meiosis 2 are that homologous chromosomes separate during meiosis 1, but not during meiosis 2. The centromeres and sister chromatids separate during meiosis 2. Follow along and take good notes as I summarize the steps of meiosis. Remember that meiosis takes place it begins with germ cells, the cells that will produce gametes. Gametes are sperm and eggs. Gametes are haploid cells. They have half the total number of chromosomes that a diploid cell has. We're going to start with one germ cell, and we're going to produce 4 gametes in 2 phases, meiosis 1 and meiosis 2. In meiosis 1, homologous chromosomes are separated into 2 cells such that there is 1 chromosome consisting of 2 chromatids per chromosome pair in each daughter cell. Meiosis 1 begins with prophase 1. Actually, before prophase 1, the cell has been in interphase. And in interphase, the chromosomes have already replicated to form sister chromatids. So when the cell enters prophase 1, there are 4 chromatids and 2 chromosomes for each of the 23 chromosome pairs. The nuclear envelope, the envelope around the nucleus, disintegrates and the chromosomes begin to condense. spindle fibers appear, which are important for the successful division of the chromosomes. To further increase genetic diversity, homologous chromosomes exchange small parts of themselves such that one chromosome contains both maternal and paternal DNA. When we look at this diagram, we see that there are two colors of chromosomes. There's red and yellow. One of the red chromosome comes from one parent, the yellow chromosome comes from the other parent. And you notice that during prophase 1, they cross over with each other. This process of crossing over is very important. It's what helps produce unique, it's what helps make each gamete unique. It's what makes this different from mitosis, where the cells are exact copies of each other. After prophase 1, the cell enters metaphase 1. In metaphase 1, the maternal and paternal versions of the same chromosome, the homologous chromosomes, align along the equator of the cell. There's a process here called independent assortment. This is also an important process that helps increase the variation within the offspring. This is when maternal and paternal chromosomes line up and randomly align themselves on either side of the equator. In turn, this determines which gamete chromosomes are allocated to, which leads to genetic diversity among offspring. So some gametes might get more of the father's chromosome for a particular set of traits, and some gametes will get more of the mother's DNA for that same set of traits from that same chromosome. After metaphase 1, the cell enters anaphase 1. In anaphase 1, each of the homologous chromosomes is pulled toward opposite poles of the cell as the spindle fibers retract. This divides the DNA equally between the two cells, which will be formed. Then in telophase 1, the nuclear envelope reforms and spindle fibers disappear. In cytokinesis 1, the cytoplasm and cell divide. They pinch off in the middle. And that results in two cells that are technically haploid. There's one chromosome and two chromatids for each chromosome. Then we enter meiosis 2. Meiosis 2, prophase 2 is really very much the same as prophase 1. In prophase 2, the spindle fibers begin to appear, the crossing over does not occur here. But other than that, prophase 2 is very similar to prophase 1. In metaphase 2, chromosomes line up in a single file along the equator of the cell. This is in contrast to metaphase 1, where chromosomes line up in homologous pairs. So in metaphase 2, you just have individual chromosomes lined up. And then in anaphase 2, the sister chromatids are pulled to opposite parts of the equator. Remember, in anaphase 1, it was homologous chromosomes that were pulled to opposite poles. In anaphase 2, those chromosomes are pulled apart so that the sister chromatids are pulled to opposite poles. Then when telophase 2 begins, this is very similar to telophase 1, and cytokinesis is also very similar, where the cell divides in two. So now, at the end of meiosis 2, we have two non-identical, well, we actually have 4 non-identical haploid daughter cells. Each cell that you had at the beginning of meiosis 2 has divided into two non-identical haploid daughter cells. So now you have four non-identical haploid daughter cells. They are all unique because they all have different mixtures of the DNA of the parents due to two processes, crossing over and independent assortment. Each haploid daughter cell contains one chromosome consisting of one chromatid. These are fully formed gametes. Compare and contrast mitosis and meiosis using the diagram on the slide. Summarize the steps in your notes. Pay particular attention to phases in meiosis where genetic recombination occurs. For example, in prophase and mitosis, the replicated chromosomes do not pair up, whereas in prophase 1 in meiosis, the replicated chromosomes pair up with their homologs, and the process of crossing over occurs. As you walk yourself through the diagram, note the differences in the lining up of chromosomes, the number of chromosomes each cell contains, and how chromosomes separate into new cells. Identify the chromosome number of resulting cells from each process. In mitosis, you end up with a 2N cell, a diploid cell. Well, actually, two diploid cells from one diploid cell. And in meiosis, you end up with four haploid cells where you started out with one diploid cell. In mitosis, when the two sets of genetic material combine, in mitosis, when the two sets of genetic material separate, each daughter cell receives one complete set of chromosomes. In meiosis, homologous chromosomes line up and then move to separate daughter cells. Mitosis does not normally change the chromosome number of the original cell. This is not the case, though, for meiosis, which reduces the chromosome number by half. Mitosis results in the production of two genetically identical diploid cells, whereas meiosis produces four genetically different haploid cells. Ultimately, mitosis and meiosis both ensure that cells inherit genetic information. Both processes begin after interphase when chromosome replication occurs. However, the two processes differ in the separation of chromosomes, the number of cells produced, and the number of chromosomes each cell contains. Recall Mendel's principle of independent assortment. Mendel found that genes that are located on different chromosomes assort independently. But what about genes that are located on the same chromosome? Researcher Thomas Hunt Morgan, back in 1910, considered the question of whether genes on the same chromosome are genetically inherited together. He studied the genes of fruit flies and found that many genes were linked in a way that seemed to violate Mendel's principle of independent assortment. As the diagram shows, during crossing over, chunks of the chromosomes, not individual genes, are exchanged between chromosomes. This means that some genes are likely to travel together on the same piece of chromosome. That is, they are linked. This is what Morgan found in his fruit fly experiments, that chunks of genes sorted independently, but genes within certain linkage groups were inherited together. One of Morgan's students, Alfred Sturtevant, wondered if the frequency of crossing over between genes during meiosis might be a clue to the gene's locations. Sturtevant reasoned that the farther apart two genes were on a chromosome, the more likely it would be that crossing over would occur between them. Crossovers are more common between genes that are farther apart. Genes that are close together are most likely to stay as part of the same chunk when crossing over occurs. Sturtevant reasoned that he could use the frequency of crossing over between genes to determine their distances from each other. This was long before scientists were able to use the high-tech equipment we can use now to analyze the location of genes. Using this logic and studying the frequency of crossing over, he was able to create a diagram like this one called a gene map, which shows the location of a variety of genes on chromosome 2 of the fruit fly. The genes are named after the problems that abnormal alleles cause, not after the normal structures. If you pick a defect here, like let's look at purple eye, find the purple eye defect on this gene map. So this gene map shows that the purple eye gene is located at 54.5. That indicates a certain position on the chromosome, on chromosome 2. So some genes are more likely inherited together. For example, star eye and dumpy wing. Look at where they are on the gene map. They are more likely than others to be inherited together because they're so close together on the chromosome. The chance that crossing over would separate them is much smaller than, say, star eye and speck body. Because crossing over occurs, star eye and speck body are not very likely to be inherited together.